Hi, good afternoon, everyone. I want to welcome and thank you all for calling in. We are excited to share with you some news about the newly approved medication for the treatment of Alzheimer's disease. It's called Aduconumab or Aduhelm. And this is the first medication approved in the last 13 years for the disease, and is the only kind in the sense that it works on the disease pathology and the disease itself, rather than just improving symptoms or other problems associated with the disease. So we put together a panel of experts who will be talking to you and addressing your questions. And hopefully you all received the letter that we sent you a few weeks ago, sharing some information about the medication and the excitement it has caused in the field, because this medication likely will lead to the release and discovery of some other more effective medications over time. So with that, I would like to hand over the presentation to Dr. Chin, and you'll be hearing from our other guests. Thank you for calling in.
Thank you, Sanjay. Hello, everyone. Thank you for tuning in. My two topics for my part of this presentation really is to go over the mechanism of action, which really means how does aducanumab actually work in the human body, and then to go over an overview of what are the potential side effects of this medication based on the findings from the clinical trials. And so I'm going to share a few PowerPoint slides with you so that you can see some images. The first one here, which I did find online, and I have the source at the bottom of this slide, really shows, and I'm hoping you can see my mouse, but that this is an infusion. So this is an IV intravenous medication that is given over a 1 hour period and is meant to be given every 21 or so days. So in essence, every 3 weeks. And how it works, it does not actually remove the protein amyloid from the brain itself, but it is actually made of human antibodies. And so the scientists were able to look at healthy elders as well as people who are older that have remarkably slow cognitive decline. And through their white blood cells, they were able to generate these antibodies that targeted the protein of amyloid in the brain. And so when you give this medication, as you can see in this little syringe, when you give this medication, it does go through into the brain and it attaches to that sticky protein called amyloid. And once it does that, it stimulates our cells in our brains called microglia, or the inflammatory cells. It stimulates them that there's something wrong. And then it's our own brain cells that actually remove the amyloid from the brain. And that is the point of the medication, to remove amyloid from the human brain. And you can see in the scans to the right of this figure, it does in fact do this. So what does it mean to have this? Well, this is a very complicated picture, and the only thing I really wanted to show you is that, and looking just at the left side, it does effectively remove amyloid from the brain. So if you do not have the medication, you are this gray line, this placebo, and so there's no removal of amyloid. But the higher dose, the purple one, this does actually reduce amyloid to near normal levels over a 52-week period. How does that actually affect human beings? Well, that's one of the things that's being debated now that it has gone through analysis and is into the scientific community. And what we find is that one of the scales that the scientists used to determine effectiveness was called CDR, which is clinical dementia rating. It is something that we do in our own research programs, and it is a reliable, validated tool. But what we found, or what they found, I should say, is that there was still decline. So to go from the bottom left across the right side of the screen, when you go up, that means you're having more symptoms, more changes. And again, that gray line is what we would see in a person who's experiencing decline. And then these other two colored lines, the blue and the purple, they're less. So there's less of that increase, which means there's still decline, but just less decline based on this sort of scale that we use. The other measures that we are looking at, some that might be more common to people, the mini mental state exam, we again see ongoing declines, but with the group that received the highest dose, there was less of a decline. This one in the middle deals with symptoms, what a person's actually experiencing. And as you go up on the screen, that does represent more symptoms. And so what we're seeing is that the people on the medication had less symptoms, but were still experiencing symptoms. The other part that I wanted to go over the side effects. And so there is this condition called ARIA, A-R-I-A, which stands for amyloid-related imaging abnormality. And what those two ones are effusions or swelling in the brain, so ARIA-E, or hemorrhages, actual small microbleeds in the brain, which would be ARIA-H. And you can see in this figure here that that white coloration, that's swelling. So there's swelling, effusions, extra fluid in the brain. And then the little dots that you might be able to see here, that represents little areas of microbleeds. And so those were two of the things that they have found with this type of therapy, a monoclonal antibody that goes into the brain. Now, what the study has shown is that there are side effects, and I only want you to pay attention to the ones in the parentheses. So this is the percentage of people who had an adverse reaction or some sort of side effect. And you see in the placebo group, the group that did not receive the medication, still 87% of people experience some form of side effect. That has to be general and inclusive. So most people experience something during a trial. But what we have here is the severe side effects. Well, again, pretty consistent between those who received the drug and those who did not. About 12 to 14% experienced something significant. But when it comes to stopping therapy, those that received the highest dose of this medication and the lowest dose still stopped at a higher rate than those that did not. And they did stop due to some of those imaging abnormalities that led to symptoms called ARIA. The other thing I wanted, actually I'm going to skip through this. The other thing I wanted to mention was that ARIA does occur in about a third of people on the highest dose, compared to only 2% of people on the placebo. So that's a pretty significant finding. It happens in people who are at higher genetic risk of developing Alzheimer's disease. So those that carry the APOE4 genetic risk. It doesn't determine that you get Alzheimer's, but it is a risk factor. And so 43% of those that had this ARIA effusion happened to be at that higher genetic risk versus 20% that were not. So those that also had that genetic risk were more likely to have symptoms. So this is really important for people to be aware of. Not only are APOE4 carriers simply at higher risk of developing Alzheimer's disease, but they're also at higher risk of developing side effects from the therapy. So it's an important discussion for people to have when thinking about considering this medication. Of those that do develop these side effects or the imaging abnormalities, they can occur within the first 8 months, but frankly, they can occur at any point, but the first 8 months are quite critical. 74% of those imaging changes that they saw in the studies were asymptomatic, meaning a person did not experience any symptoms, but they were identified based on the surveillance, the imaging that happens throughout the study. And then of those that did develop symptoms, most of them were quite mild, 67%, so 2/3 of people, but some of them were significant, and we have to be on the lookout for those. The most common symptoms related to the swelling or the microhemorrhage would be confusion, dizziness, nausea, even some visual changes. They do typically resolve within 4 to 16 weeks, but there is a protocol for stopping the medication and even determining whether to restart it based on the clinical trial. They're very rigorous in clinical trials, and so that's something that's going to need to be considered in the clinical world as well. So I'm going to stop there and turn it over to Dr. Gleason, who's going to talk about a really important aspect of the clinical trials.
Thanks, Dr. Chin. So for those of you who don't know me, I lead the Inclusion of Underrepresented Groups core. So my career, my life's work is focused on the inclusion of groups who are affected by this disease, but who are unfortunately underrepresented in our trials looking at treatments and prevention strategies. And my partner in this work is Dr. Fabu Carter, and I will open the floor to her as well. But my take-home message from this brief conversation I'll give you some data to support this take-home message. We've got to do a better job with inclusion. By not including a full and diverse group of participants, we don't have the full story about the effectiveness and the safety profile for all groups. And this is actually, as I mentioned, important, and there are data to support cases where this did not happen, and there were results that led to inequities or disparities in how treatments work. The other important message is that clinical trials are beneficial for people. Even if you're given the placebo, there is something called the placebo effect, where people benefit from believing that they're being treated. Also really important. The chance to be part of cutting edge research and maybe get access to treatment before it comes to market. So I wouldn't be doing this work if I didn't believe in the importance of clinical trials, and that this is important that we are inclusive in our research. Now, some data as to why we need to do a better job. You notice in Dr. Chin's presentation, there were two titles in there, the Emerge and the Engage studies. So those were the two trials that supported the FDA application for approval. So there were about 3,300 individuals enrolled in that trial. Interestingly, in the Engage study, we don't actually know the full breakdown of race and ethnicity. We have only a report from the paper as to how many white participants and how many Asian participants there were. In the Emerge trial, with some digging, we were able to find some report out on the number of individuals who were non-white, and it's egregiously small. I'll put it that way. Only 0.7% of that study population, 1,600 individuals approximately, were Black. Only one individual is American Indian. The vast majority, including, were either Asian or white. And the reason that breakdown, how they describe it, is because of where they were enrolling study participants. It's mostly European, Northern American, Australia was included, and a handful of Asian countries, including Japan, South Korea, and Taiwan. For the most part, we are looking at countries where there are a lot of resources and a lot of engagement with the scientific community. So hopefully my take-home message that we need to do a better job, that comes through loud and clear with the few people that we have enrolled in these studies, and an example of when this has gone wrong. So most people in the Black community know about the story that emerged, the headline that emerged, that the HPV vaccine was less effective in Black women. And the manufacturers of that drug, that vaccine, were very honest, basically saying the reason we know this is less effective with Black women is because we didn't include enough Black women in the trial. So in the meantime, this treatment, then there's a disparity in efficacy because at the front end in the development of the drug, we did not do our due diligence as scientists in developing this particular vaccine. Let's not make the same mistake with our treatments for Alzheimer's disease, especially considering that Black and brown people are more likely to be affected by the disease. So I'll yield the rest of my time to Dr. Carter.
Thank you, Dr. Carey Gleason. So I am a senior outreach specialist / program manager for recruitment and retention. So the good news is that in our studies, we have 25% of the total population involved in the study. And as Dr. Gleason has pointed out, regrettably, we can't say the same for this new drug, that the percentages aren't there of people of color, specifically African Americans and Native Americans, to feel comfortable and to feel good about recommending it. There would be some hard criteria that you would have to think about first. So for example, I personally have a family member now who's experiencing memory loss. And looking at the drug and looking and listening to everything that says, 0.7% does not reassure me about how it would be effective for her. So whether it would be better for her to take it or better for her not. And one shouldn't have to really think about these things as we're pointing out. There should have been a large enough percentage for me as an African American, or if I were a Native American, to say, this has been tried out on populations that are specific to me. And I can say with confidence that this will do no harm. It will be better for me than worse. So it's a practical analysis. It's a people analysis that the people, for example, African Americans who are leading in the disease, the fact that they weren't included in larger numbers in the study for this new drug leaves our community as the Black Leaders for Brain Health once again searching for answers and once again wondering why the drug could come on the market without having adequate populations involved in the research.
Thank you, Dr. Gleason and Dr. Carter. I think we'll move on then to Dr. Carlsson, who will talk about future clinical trials and the impact on these studies.
Thank you, Dr. Chin. So again, I really appreciate Dr. Asthana's introduction, Dr. Chin's introduction about the new drug, and then Dr. Gleason, Dr. Carter's input on how important it is for inclusion of diverse individuals in these studies. So again, I think, Dr. Carter's point about we care about the people outcomes is really important, because in the past, these types of studies would focus on what does the medicine do on the brain or on these certain questionnaires or certain cognitive tests. And, I think a lot of us who have family members with dementia or at risk for dementia, what we really care about are the people outcomes. So how is this going to impact my life, my ability to think clearly, to be able to be functional? And so over time, these clinical trials have started developing kind of two paths. So one, they want to see, do these medicines work like we think they're working? So do they bust up the amyloid plaques or trigger our bodies to bust up the amyloid plaques? But also, do they help us think better and function better, enjoy our time with our grandkids, et cetera? So again, those things together, I think that the encouraging part is that clinical trials have started to measure that better. So that's a positive change in these clinical trials. And that's where a lot of the controversy is with Aducanumab, because they're trying to see, well, it looks like these cognitive tests or these thinking tests improved. It looks like the drug did what we think it did, but how much of an improvement is it for the person? And so that's what a lot of the controversy is being weighed right now. But the point about representation is really important too, because in addition to being a researcher, I'm a clinician. So I'm a physician who's going to be prescribing Aduhelm down the road or another drug similar to it. And what I tend to depend on is looking at those studies and seeing who are the people treated in that trial. Do they look like my patients? So if I want to be careful about those side effects and those risks, I'm really going to probably restrict who I prescribe it for people who look like those people in the trial. So that means people in a certain range of cognitive test results, people with certain health conditions. So if I have, let's say, an African-American participant who maybe has some vascular risk factors that weren't included in the trial, then I may not feel as comfortable prescribing it. So again, that really limits who the drug is prescribed for. So that's why it's so important. And so we have clinical trials in our program. So we're launching a study called the AHEAD study that's looking at an anti-amyloid therapy in people who don't have symptoms yet. So it differs from the EMERGE and ENGAGE studies. They don't have symptoms yet, starting anti-amyloid therapy. And there are a lot of other trials. I have no financial investment in the drug being used in AHEAD. There's a lot of other trials going on, but it's really important for these trials to include people who are from underrepresented backgrounds, African Americans, Native Americans, people with Down syndrome, and these other conditions that put them at risk for developing dementia, so we can better understand how do these drugs work in the patients who are going to be coming to me, asking me as a clinician, is this drug good for me? So again, I think it's this approval and the controversy surrounding it has really, really emphasized that we need good clinical studies, good clinical trials that include the people outcomes, like Dr. Carter mentioned, and the markers to see if the drug's doing what it's supposed to, but also diverse representation so we can make sure after it's approved that we can prescribe it to the people who need it.
All right, thank you, Dr. Carlsson. And so for the remaining time, I would like to open it for questions to be asked from our Facebook audience. I have some prepared questions that were sent to us in advance of this, so thank you for those of you who were being extra proactive. The first question, when does a person start treatment with this medication? Is it at the onset of the first symptoms? And Dr. Carlsson, since you already started talking about being a potential prescriber, I'll direct that first one to you. Oh, I believe you're muted.
Oops, oh, I'm muted. Sorry, I was just say I have to refer back to. So again, these are the people who are included in this trial, and I'll have to, someone can jump in and say which, there are a certain range of cognitive scores. So these are people who do have symptoms. So that's different than people who have identified to have amyloid buildup in their brain, who don't have symptoms yet. These are people who do have diagnosed mild cognitive impairment or mild dementia. So what that means is that people with moderate or severe, not just dementia, but Alzheimer's type dementia are not included in the trial. And people who have amyloid buildup but no symptoms yet are also not included in the trial. So it's dementia, but it's a subgroup of dementia, the Alzheimer's, amyloid, and then in a certain range of cognitive tests. So I think they're based on many mental scores, and I can't remember the exact range at baseline, but it's a certain narrow area.
Thank you, Dr. Carlsson. And now I forgot to mention that Dr. Sterling Johnson is here with us as well. Dr. Johnson, please turn on your camera and show our audience your face. And the next question is for you. How much brain amyloid plaques or tangles appear in the brain based on their stage, whether you are someone who does not have symptoms, has mild cognitive impairment, or throughout the spectrum of dementia?
That's a great question, Dr. Chin. There's a lot of variability, but by and large, people who have Alzheimer's disease have quite a bit of very unmistakable, very noticeable amyloid plaque in their brain. And when I say Alzheimer's disease, I mean people who have dementia, people who have MCI, and even people who don't yet have symptoms. There's an unmistakable, unambiguous amyloid signal in the brain. And this drug and others like it that are coming down the line are very effective at reducing the level of amyloid that's in the brain.
And the next question actually relates to this. Does the drug work all over the brain or in specific areas where there happen to be more amyloid?
I can answer that one as well. It works, it works on the amyloid plaques itself. So it'll work wherever the drug detects those plaques are occurring. They happen to be in the cortex of the brain, which is the outer part of the brain where our brain cells are. We call it the gray matter. And that's where we see the most noticeable effects with this drug.
I have a question from our Facebook Live audience. What is the end game for a prescription with Aduhelm? Does it merely reduce the amyloid for a period, or is long-term prescription going to be needed? Dr. Asthana, do you, as a potential prescriber?
Yeah, thank you, Dr. Chin. That's a great question. And I think this is something that we still don't know a definitive answer. So just to point out that the FDA, when it approved this medication, the approval is conditional. And the condition is that Biogen, the company that produced this medication, has to continue collecting data on people who are getting the treatment about how their cognition is, how their memory tests are, and how their day-to-day function is. So the answer to the question would be, as we learn more about the longer-term effects of this medication, that's when I think we learn when to stop or how long to continue. The treatment trials were only for 18 months. And within that duration, there was no definitive evidence of improvement either in memory or function. So the longer term follow-up that the FDA has mandated on Biogen, that people who receive the treatment should be followed for a number of years. And then Biogen will report back what the effects on memory and function are. And until we know that, we don't quite know how long this treatment will be given.
Thank you, Dr. Asthana. Dr. Gleason and Dr. Carter, how do we improve participation from people living in underrepresented areas who are from underrepresented backgrounds?
Oh, thank you for that question. I think it's incumbent upon the scientific community to do a better job and do a better job of engaging and making sure that this is a trusted institution, a trusted project for people to participate in. And we do that by building relationships. Absolutely essential to do that. We have lots of models. There are a lot of people working in this space, and other groups have done a better job of engaging. Like the cancer researchers, for example, have been more successful in engaging groups underrepresented in research. And I think in the Alzheimer's disease world, we just need to catch up with that. And really, it's on us to make this a something that is safe and inviting and culturally appropriate. So Dr. Carter, do you have anything to add?
Well, we all know that this is a very dread disease. And of course, the people who suffer the most from this have a really important interest in seeing interventions and medications and a change in how this disease is treated. So working on that premise, and again, thanking people who are in our current study makes us believe that the same mechanisms we use for recruitment and retention can be utilized on a larger basis, but also that can be used for every study. Every study has the potential to, to have all people involved in it. And so one of the discussions, again, in Black Leaders for Brain Health that has been working with us is that, they would like to be at the forefront of making sure that not just African Americans and Native Americans are recruited into the research, but who and what they find when they are part of that research.
And if I may, Dr. Chin, I'd just like to add that at the institution level, one of the top priorities for the ADRC, for UW-Madison and UW Health is to increase the diversity of our studies that are being done in Alzheimer's disease. And thanks to Dr. Carter and Dr. Gleason and their team, we here at the UW-Madison at the ADRC are extremely successful, although a lot of more work has to be done in enrolling people into our studies. In fact, our ADRC is among the best performing ADRCs in the country in this area. Nevertheless, we need to make significant improvements by enrolling more people from diverse communities into our studies. So we want to encourage people to think about it and also thank Dr. Carter and Dr. Gleason for their superb job. Thank you.
I think to end, we might have two questions here, one from our Facebook Live audience. How much does Aducanumab cost? And our insurance company is going to cover some of those costs. Right now, it's listed at $56,000 per year for the infusion. That does not include the costs of the infusion center, the healthcare provided, and the monitoring, which would require MRI scans of the brain, as well as confirmation that you have amyloid in your brain. So that's really an estimate of just the drug itself. The next question is a very important one that's being explored is, will insurance companies cover this? It starts with Medicare and at the national level to determine appropriate use and coverage. And that is happening now. And that will lead the conversation going forward as to what will be covered and how much of it will be covered. But we don't have an answer right now. The last question is a two-parter, very interesting. If the drug adheres to cells and stops the plaques from building up, where does the plaque actually go? I don't know the answer to that one, so I'm going to look to my colleagues.
Yeah, I don't know if we know the answer. Dr. Johnson, would it eventually be excreted or cleared through the blood? It'll cross the blood-brain barrier and
Yep,
the blood and be cleared?
that's right. The plaque accumulation is it's a clumping of these proteins, and the immune response by the brain cells dissolves that clump, and they are the proteins that are left are just discarded in the brain's usual way of getting rid of metabolites and waste products through its own little system of getting rid of those things. And so I think that's how this works.
And the last part of this very interesting question, I'm assuming the drug does not stop the plaque from developing. What happens there? And I think that's a really important point, and that's why I'd like to end with this, that the framework that scientists are using, this amyloid tau brain cell death, which we often refer to as ATN, it is a framework. It is pathology. Amyloid is in the brain, and we do believe that's a part of the Alzheimer's disease process. But that doesn't mean, that doesn't explain all the potential reasons why someone could have amyloid, why it progresses, why tau forms, how that leads to the symptoms that we may experience, which is why research in this field is so critical, which is why we rely on our participants to come in and do the tests and give their time and parts of their body so that we can really understand that question. Because we need to be able to figure out what drives all of this. And most importantly, what can we do? Whether it's a medication or more, from our perspective, our daily lives, what can we do to reduce that buildup of amyloid, or whether or not it is tau or whatever the next protein is. And so that is an important question that we're trying to answer. But we do that because of participants and people interested in the community, like those that are watching. So with that, I'd like to thank our panelists here, our experts in the field for their time and being able to answer those questions. And for you, our audience members, thank you for tuning in and asking your questions. We are still available, so feel free to send messages on Facebook or to email our center. We are here to answer questions as we are learning the answers ourselves as this process occurs. Thank you, everyone.
Thank you all for
Bye.
participating.